Hello, my lovely anatomists and physiologists. Michele Glass here. This is exactly where we left off with our last video, talking about the 3 phases of hemostasis or stopping blood loss. We started with the vascular phase, which is immediate response, very soon followed by the platelet phase, where we get that platelet plug. And that's like our temporary plug or stopper. And then we get those platelets releasing all sorts of important chemicals that just kind of keep this sequence of events going. This whole process, remember, is an example of a positive feedback loop. And then we left off with our coagulation phase. And spoiler alert, we said that the coagulation phase ends with our blood clot. So let us talk about that coagulation phase in more detail. The key thing that we're going to see with this phase is we have what is described as an enzymatic cascade. And a cascade is like a series of steps where you should think like step 1 gives permission for step 2. Step 2 gives permission for step 3. Step 3 gives permission for step 4, and so on and so forth. And so the beautiful thing about enzymatic cascades in the body is that it's got this whole built-in series of checks and balances. Remember, spoiler alert, we're going to end with a blood clot at the end, right? So let's just write that down. We're going to get a blood clot. A blood clot is a beautiful thing if you have damage in your vessel, right? If you've cut your vessel, you need to stop your blood loss. That's the blood clot. It's beautiful. It's wonderful. It keeps you alive. It's a really bad thing if it's just spontaneously happening and then it's causing a barrier in your blood vessel. And now, instead of keeping you alive, it's doing the opposite, right? Making life hard, making life over. And so we do not want blood clots to spontaneously show up, right? So that's okay. Let's put in a whole bunch of steps, a whole bunch of checks and balances. Let's make it so that you have to have like permission essentially for each step. And so let's begin. You have these 2 different pathways, and you can see that they both lead to what we refer to as the common pathway. Now, in the case of your intrinsic pathway, this is initiated by internal damage. Internal damage to your vessel wall. We're going to see that this is the longer, meaning few minutes, more complex. And we're not going to study the details of each step here. Okay, so we're going to study the details, each step of the common pathway. But we're just going to be very, very, very general in our descriptions of the intrinsic and the extrinsic pathways. You're welcome. When we talk about the intrinsic pathway, what we will see in terms of our steps is you're going to be getting clotting factors from those platelets. You're going to get platelet factors, again, from those platelets. And then you also get some signals from those endothelial cells. So those are those cells that are lining that blood vessel. And that's going to lead us to the common pathway. When we look at the extrinsic pathway, we see that this is coming from trauma to extravascular. So extra meaning outside of the blood vessel. Extravascular cells. And here we see that it's much quicker. So now we're talking like seconds instead of a few minutes. And it's a more direct pathway. Now, depending on the trauma, you probably have intrinsic and extrinsic pathways happening simultaneously, right? So if you have a full cut through your vessel, you would expect internal damage, but you would also expect some damage to the cells around. In this case, we're getting clotting factors and we're getting the release of calcium ion, which we mentioned is coming from our platelet cells. And that's leading us to our common pathway. Now, when we talk about this whole function of the coagulation phase, we're going to need calcium from our diet stored in our bone, right? And we're also going to be needing vitamin K, which we can get from our diet, but we also see it produced by the bacterial buddies that live in our large intestine. So I'm abbreviating large intestine here, LI. I did that earlier in an earlier video. Okay, so what's happening? The intrinsic pathway and the extrinsic pathway both take us to the common pathway. And what we see is that all of these different clotting factors have been described by a name and a number. They're all listed for you in your OpenStax book if you want to read about those. We're interested in factor 10, and they're assigned the Roman numerals. So this would be factor 10 with that factor X, right, for a Roman numeral. So our factor 10 is what we're getting to at the end of the intrinsic pathway. We get to factor 10. At the end of the cascade for the extrinsic pathway, we get to factor 10. So we can say that factor 10 is the beginning of our common pathway. And once you have your factor 10, that's going to signal this enzyme prothrombinase. And we can, of course, recognize it's an enzyme because it has that ASE ending. And when we have this enzyme, we're going to get an inactive prothrombin converted to thrombin. Okay, so this is again talking about permissions, right? Step 1 has to happen before steps 2. So the entire intrinsic pathway cascade, all those steps have to happen to get us to factor 10. Or all of those steps have to happen in the extrinsic pathway to get to factor 10. Factor 10 activates the enzyme prothrombinase, and prothrombinase has to be present in order, maybe I should draw it that way. Prothrombinase has to be present in order to convert this inactive prothrombin into our active thrombin. And when we have our active thrombin, then that's going to convert That's going to convert our fibrinogen into fibrin. And once we have fibrin, we have our blood clot. Okay, a couple things. Fibrinogen we talked about as one of our plasma proteins. It's always in circulation. So again, this goes back to like, we don't want to spontaneously convert fibrinogen to fibrin. We want a hell of a lot of steps with a lot of controls to keep that from happening. So that's why we see a cascade to get us to this step. Fibrinogen is soluble, meaning that it's floating through the bloodstream. Fibrin is insoluble, meaning it's a strand. It's a protein strand. And what we see then is that protein strand is going to trap all your platelets, it's going to trap all your red blood cells, it's going to trap all that stuff to make the blood clot. So the blood clot is like platelet plug plus fibrin. You can think about fibrin as like the webs of a spider web, right? It's those strands, those sticky strands that's holding the whole thing together and trapping it, right? And so this is making that beautiful blood clot that we want to have when we've damaged our vessel, but we don't want to spontaneously have the clot. Okay, ultimately, we're going to be repairing this vessel. Ultimately, we have to remove this clot. And so then we have a process called fibrinolysis, right? So lysis means to break apart, and in this case, we're breaking apart the fibrin, and the fibrin is that sticky web, right, of our blood clot. And so this whole process we can say is a gradual because we want it to do its job, but then we need to get rid of it when we don't need it anymore. So it's a gradual degradation of the clot. And it's also a set of steps. It's also a cascade. So here I'm just going to say it's a cascade. We're not going into all the details. Ultimately, you get an inactive plasminogen converting to an active plasmin. And it's this active plasmin that's going to actually then dissolve that blood clot. Okay. So remember, with like any kind of mechanism in the body, we have to like take care of the emergency type of thing, but then we also need to return to homeostasis. All right. Up next, blood typing. So stay tuned. As always, take care of yourselves and each other.